It's a privilege to be a colleague of Andrew's as well, and it's a privilege to be here. I have to say, I hope I can rise to the standard that the talks have shown today. I've learned so much and actually incorporated a number of the talks into what I'm going to talk about tonight because they're so relevant. The other thing is the Save the Children motif is something that I didn't know was going to precede my talk, but I'm actually going to close out my talk with a reference to that. And then finally, the $3 billion project that my team ran and came in ahead of schedule and under budget. If it were my $3 billion to spend, I probably would have spent it on a couple of other things, but I had the world's best team who did that, and full credit goes to them for that success. So I like to look at things, large questions from multidisciplinary angles, and ask more questions than I give answers. So a lot of what I'm going to talk about now is questions rather than answers. So if we could go to the first slide, please. And I have a number of really bad habits. One of them is really long titles for my talks. The other is making up new words. There's about a dozen words that if you Google them, you find my name. And a couple of them are in here. But the title of the talk is The Future of Global Collaboration as the Panorome and the Pleco System Go Exponential. So I'll explain what the Pleco System is in a bit, and our obligation to children of the sci-fi generation. And by the sci-fi generation, I mean that any child born today, if they can imagine it, they will see it happen in their lifetime. That is what's happening with the exponential rate of change that we're experiencing. So starting off, this is the audience participation phase. So a series of questions here. How many medical informaticists in the crowd? Show of hands, please. Okay, handful. How many geneticists? Another handful. How many clinicians? Wow, okay, and how many analysts? And folks involved in policy. Cool, great mix, okay. How many have had your exome sequenced? Your full genome sequenced? One exome, no genomes. Wow. 500,000 in the UK and not one in the audience. Okay, gotta walk the talk, guys. Then I want to ask you how many have encouraged your family to do it. When you get your genome sequence in the next week or two, you should encourage your parents, your brothers, your sisters, your children to do so, because the real cool studies going on right now, like the one that Google's doing with autism, include trios. Index case, mother and father. Why? Because we know that a lot of genes and alleles that tend to have adverse consequences or significance in some people have no significance whatsoever. Best way to figure that out is to look at your parents, your brothers, your sisters. So get sequenced, get your family sequenced. How many have heard of either Sage Trust or the Genetic Alliance? Anybody? Okay, oh good. And these are two organizations that I'll come back to a little bit later that are involved in citizen donation and control of their own data. How many have had your microbiome sequenced? Okay, a couple, good, okay. So I get 4 microbiomes sequenced at Stanford every 3 months, and anytime I get sick, usually after some foreign travel. And how many have heard of CRISPR-Cas9? Show of hands. Okay. Anybody want to tell us where CRISPR-Cas9 comes from? Anybody know? It's an immune defense mechanism in primitive bacteria, the Archaea phyla of bacteria. It's sort of a memory of offensive plasmids and phages. And this immune system mechanism in bacteria has now been applied as a gene editor. So all of you know that in databases you can create, revise, update, and delete. This complex from bacteria has been used very successfully, very globally, to do gene editing of those 4 functions. So it's like a database editor. Anybody heard of Gene Drive? Anybody know what Gene Drive is? Okay, it was pioneered by Austin Bird at Imperial College. And basically what it does is it uses CRISPR to say, I want to eliminate all alleles but this one allele. So it can go into any genome in any cell, and this is actually proving to be very easy in plant or bacterial or animal or human species, and use one copy of one allele to copy itself on the other and obliterate the other allele. What it basically does is it drives alleles to extinction within a population has been proposed, for example, as a way to render all mosquitoes and all malaria in the world incapable of infecting humans. A lot of great uses, but you can imagine a lot of not so great uses that can come from that. It's only recently been applied, and there's already an international agreement about restricting how it's used. We can screen today for 500 inborn errors in metabolism in embryos, and how many more might we discover soon? Lots. And what about polygenic influences? It's been predicted that the DNA storage needs on the pathway that we're on today are going to exceed that of YouTube by the year 2025 at 1 to 2 exabytes a year. It still turns out that DNA is by far the most efficient and compact mechanism of storage. You can store an exabyte of data, 10 to the 18th bits of data in 1 gram of DNA. And a friend of mine, George Church, worked with George Church at Harvard, and they encoded an entire book, not coincidentally, that is about biomimicry and how technology mimics biology. They encoded the entire book, including graphics, using DNA. And what percent of mothers carry stem cell clones in their children? Does anybody know? Any guesses? 100%. So if you are a woman who's carried a child to full term, you have stem cells in your organ systems from each of your children. Pretty fascinating implications for that. Do you know how closely related you are to your Facebook friends? This has been studied genetically. This is James Fowler at UCSD and Nicholas Christakis at Harvard did this study. And they found that on average, your friends on Facebook are as closely related to you as if you shared a great-great-grandfather. This is like amazing. So they say you can pick your friends but not your family. Well, kind of, sort of. There's a little bias in there. So do you know why $500 billion was spent last year on pharmaceutical company mergers and acquisitions? That's $500 billion. They understand that in the world of personalized medicine, the cost of drug development is not going down. The market share is going down. So as we develop more and more personalized meds, they have fixed development costs and declining market opportunities. And they're undergoing a massive consolidation today. So they're way ahead of us in terms of understanding the need to go global. So there is a financial and economic model illustrating why global collaboration is critical, evidenced by the pharmaceutical companies. Has anybody read the book Bad Pharma? It's written by someone from the UK. And it basically, among other things, talks about publication bias, where if you fund 20 studies and only one of them shows a benefit from your, as a pharmaceutical, a benefit for your drug, you publish that one and forget to publish the other 19. Well, think of what this consolidation means for the future of publication bias. Just a couple days ago, they announced that there's a universal influenza vaccine. Rather than attacking the hemagglutinin head portion of the antigen in influenza, which is highly variable and why we have to have a new vaccine every year, they attack the stem portion of that hemagglutinin antigen, and there's hope, and there's human trials starting soon, that we can have a vaccination for all influenza, including H1N1, H2N5, and so forth, using this new technique, clearly with a genetic foundation. And then one of the terms I've coined is Metcalfe's law. So Metcalfe's law says that as you add each node to a network, you bring more than linear value. So if it's node number 100 added to 99, it adds more than 1% value to the network. So my version of that in the corollary, and I hybridized our last names to Metcalfe's law, is that when you begin aggregating data around an individual, their phenome from their health record, their genome, their microbiome, their exposome through multiple sensors that they wear, and so forth, each additional data type that you add, you bring about greater than linear growth in the value of the pre-existing data points as you begin to understand this. I'm going to talk a little bit more about that later. But one of the concepts, and the same is true for institutions, the more collaboration between institutions that are working together, the more disparate data types you can bring together and create a better understanding. So who cares if somebody in Sweden has 100,000 EKG tracings and someone in South Africa has 100,000 microbiomes? If they're not linked by the individual, the value of those disparate data are essentially siloed and isolated. But when you do bring all those data together, it creates something that's become known as the Mosaic Effect, and that is you simply can't de-identify data when you start bringing together all these data sources. So the notion of de-identification, which is still being bannered about everywhere and legislated in the US once again, just recently, is a completely obsolete, quaint notion in the world of big data and data aggregation and the world of little data, where we bring all data together from different individuals. So what are some of the big looming questions that we need to think about? So how do we facilitate global collaboration in a world of diverse and controversial privacy policies? There's a couple of mechanisms that are emerging right now. One, the Global Alliance for Genomics and Health that Andrew and I co-chair, the eHealth work group. We're looking at a concept called data enclaves and credentialed access. So what you do is you don't allow people to have your data, you allow them to query your data. And the second is this model of citizen donor control. So I mentioned before Sage Truss and John Wilbanks and Genetic Alliance and Sharon Terry, and what they do is they aggregate data on behalf of individuals and just store it. They do nothing with the data except store it for the individual and operate as a consent distribution model, or as Sharon Terry calls it, a switch in a network, where if the person says, I want my data to go to this study and this study and this study, but I don't want it to go to that study, they just simply execute the requests of the individuals. So the other second question is, why must these panoramic studies that go across all these data types span international cohorts? Well, earlier today, Kathy Sudlow mentioned that the UK Biobank consists of 95% white members and that they have 50% the morbidity of the average citizen of the UK. And she made a point of saying, we know that this is not representative of the general population. We do not extrapolate or presume anything about prevalence of disease based upon this voluntary cohort. So the implication of that is the larger the sample size that we access in any of our research for any purpose, the more likely it is that we'll have durable results. And John Ioannidis is a professor at Stanford who has shown that almost half of all published studies in referee journals in the medical field are subsequently disproven, and many of those that are not formally disproven are actually discarded as obviously wrong. So durability of data is important. There's a book called The Half-Life of Facts by Daniel Arbison, published in 2012, that I encourage you to read, that indicates in web world, we're seeing the half-life of facts get shorter and shorter and shorter. So how do we use big data to expand the population so that we're not getting artifactual or arbitrary results that will stand up to the test of time? Second example of this is yesterday Gil Alterovitz was here. He had to go leave today to go speak at Oxford tomorrow, but he's done studies on TB resistance from 20 different countries while at his lab at Harvard. And what he's found is that there's an associated protein and genotype in the bacterium that, when identified, is a very, very strong predictor of multiple resistance to antibiotics. So think of the implications for that vis-a-vis one of the talks just given this afternoon on screening for TB upon reentry to the UK. So Baruch Spinoza, a great philosopher, spent his career looking at the trade-offs of virtues. So privacy is a virtue. Population research is a virtue. Increasing medical knowledge is a virtue. But we all know that they come into conflict, otherwise this whole privacy issue would just go away. And so he spent his life really looking at trade-offs between autonomous virtues that come in conflict. So how would he cast privacy versus knowledge generation in the world today? And question, how many of you have heard of Bitcoin? I assume most of you have heard of Bitcoin. How many of you heard of blockchain? Okay, so blockchain is the technology underlying Bitcoin. We're launching an initiative, and Rodrigo Barnes, among others, is helping move this along, and some folks at UC Santa Cruz. But that same technology that underpins blockchain may well be a mechanism by which we can provide a level of security and privacy, and at the same time do microcredit accounting. So Bitcoin is microcredit accounting with low friction, almost no friction. And so there's a open source not-for-profit group called Ethereum, which is helping apply that technology in fields outside of finance and monetary Bitcoin approaches. And we're seriously evaluating the possibility of being able to use that same technology in healthcare to help address some of the conflicts between maintaining privacy and being able to say, Angelina Jolie, on a weekly basis, thank you so much for donating your entire genome and your entire phenome to science. As of this week, you now have 22,000 women who have benefited from the research done on your allele of BRCA, the gene that predisposes to breast and other cancers. And likewise, do the same for researchers. Today, your research was reused in 27 algorithms and 15 decision support systems. So microaccounting is an area where we may be able to use that. Another question is, what's the role of reciprocal transparency? So people, and there was a great talk panel earlier today about trust and how you generate trust. I believe that one of the key elements of trust is reciprocal transparency. So we trust researchers we entrust them with our data, but we trust them to take good care of it. What if we have transparency into how our data is being used and who's using it for what purposes? And that reciprocal transfer, there's a great book called Social Transparency written in 1998 by David Brin that I recommend that sort of predicts the future of social transparency. How does power analysis change in big data analytics? So those of us that are conventional analysts working in conventional databases, we can calculate how many members there must be in a cohort to reach a certain level of confidence that a difference is truly significant in a treated or untreated group. In the world of big data, we're using massive arrays of data types. What does power analysis mean, and how do you validate what is significant? A related question, which institution is going to grant the first PhD in what I call meta-analytics? And that is, what sequence of analytics is best suited to which question on which data substrate, and which ensemble of analytic tools is best for which study. So there's a whole array of analytics that's just emerging now. There's a thing called data interrogation, where when you have a finding in the world of big data, how can you interrogate that data source in different ways to cross-validate that it's not a spurious finding? And then finally, how does does the human civilization require a diedarity? That's another one of the words that I made up, to monitor the singularity. So the singularity refers to how everything becomes one. Machine, human is all part of the same gamish technical term. My notion of the diedarity is, and Elon Musk, founder of Tesla and SpaceX, and Stephen Hawking, the famous physicist, who I'll refer to again in a moment, have said basically that AI and robotics really does threaten the human species today, because as soon as we can create one instance of AI and robotics that is as competent as the smartest human, they can then collaborate in mass ad infinitum and bring their knowledge and ability to influence outcomes, rapidly racing ahead of the human race. Others, like Peter Thiel, have said that they're worrywarts and doomsayers, but in fact, I think the truth is somewhere in between the two of those. And my notion of the diedarity is that we need to have humans that apply new technologies in AI to ask three questions of the singularity as it emerges. If something evil is going on, what would it look like? If something evil were going on, how would I find it? And if I find it, what do I do about it? And I think those are going to become very important questions of the future, because I believe that what Stephen Hawking, not a amateur scientist, and what Elon Musk, a very accomplished entrepreneur, have both called out as serious risks of the future. I gave a talk about 6 years ago called Who Owns Your Avatar and Why Do You Care? And so if you really look at what we're after in healthcare, we're after motivating people towards healthier habits. And there are literally scores of startups that are using avatars today to help motivate each of us to reach our own goals, or so they propose, and so they probably are today. But it's my belief that those avatars will become so sophisticated that they will be owned by large Fortune 100 companies, and that they will provide them for free. Why? Because they can insert whatever. I believe it's going to be the most effective channel of marketing ever to promote various behaviors that may be associated with a purchase of particular products and so forth. And so who owns your avatar and what transparency you have into that avatar, and what are the messages that are not the ones aligned with your stated values and objectives. So Lee Silver, the co-founder of GenePeaks, which I'm going to show a video in a second, has said that sex is a really dangerous way to reproduce. So if we could cue up the video, I want to show something that, now I promise it's not X-rated. This is from 60 Minutes. You go to full screen after you get it going. So what they do is, in vitro fertilization, they generate 4 fertile embryos. They do the complete genetic sequencing of all 12. They look for 500 inborn errors in metabolism, including major diseases. And they go ahead and pick the embryo that is not affected by BRCA. So this is clearly something that for this family is of profound value, but it also raises the question of, well, we've got 6. Half of the, statistically speaking, half of those 12 embryos are not going to have the BRCA gene, but one of them has blue eyes and the other has green eyes, and the other one has a height of 6'2" and the other has a height of 5'4". So this is, there have been over 2,000 children born by this technique today. Okay, so this is not futuristic, this is today, and it's already bringing us into the brave new world of eugenics, and invites all kinds of ethical concerns. I'll just say my own view is, as long as we can help this mother have children without the BRCA gene, I'm okay with it. If they want to pick blue eyes, that's fine. But there are other implications that are more worrisome. So I mentioned I'd come back to Stephen Hawking a little bit. So Stephen Hawking did his initial theory on black holes and how the Big Bang of the universe originated in the ultimate black hole. And there's been an exponential accelerating dispersion of all space and all matter over all, all energy and all matter over all space and all time. And I like to think of big data analytics as the exact inverse of that, where all data and all knowledge is accelerating into a single point in time, and is allowing us to have a much clearer, deeper, broader view into what reality is that we live in. The fascinating thing is that just in the last couple of weeks, Stephen Hawking has revised his theory about black holes and has said that basically a lot of the information that was in that original black hole and a lot of the information that happens when a galaxy collapses back into a black hole is actually retained in holographic or parallel universe constructs. So it's fascinating that there may be lots of symmetries here, but we're on the symmetry of collapsing data and knowledge into a point in space and time. So does anybody recognize this very, very famous piece of abstract art? My wife and I saw this at MoMA in New York City and laughed at it and said, that's the stupidest thing I've ever seen, until I read a book called The Invention of Abstraction, and it explained why this was so profound. So this is by Malevich, done in 1915. This is when Picasso and others were inventing abstraction. And what was happening at the time was that Picasso had made an assertion that abstract art was only valid to the extent that it retained a thread of linkage to reality. And Malevich basically said, you know, fool on you, this is a boy with a knapsack. And so this was actually breaking that constraint that Picasso had put on the world of abstraction. And the reason I bring it up in the world that we're in with big data is this is the social network that Malevich and Picasso and Claude Debussy and a whole cohort of poets, authors, artists, and musicians were collaborating in bars throughout Europe at the time. And it was the net result of this interdisciplinary collaboration that resulted in the invention of abstraction and ultimately breaking completely the connection with reality. So I think there's many parallels, and this is almost exactly a century ago when this happened, with what's happening today with big data, taking the multidisciplinary look and the beginner's mind and throwing away your constraints and your assumptions and looking for new hypotheses that have never been thought of before. So a lot of people really don't have a grasp of what's different about big data. It's not just more data. It's not just more computing power. It's a very different world that is less constrained by data models and database schemas. So in a conventional database, you really want to put round pegs in round holes and square pegs in square holes. In the big data world, you basically just want to represent all the data. And it's been said that the big data world is optimized for generating hypotheses. So you use visualization tools and you see what shows up from very disparate data types, and you start seeing relationships that you didn't even imagine. You didn't ask the question, is this related to this? You just did a mashup of all these data types, used powerful visualization tools, and lo and behold, look what you find. And then you go about to cross-validate and see if that's real. So that's as opposed to the conventional model. You generate a hypothesis, you test it in your database with a database schema where every peg has a hole that it fits in. And so this is a very, very significant distinction between the big data and the conventional data analytics worlds. And they both, in my belief, will coexist for a long time, even as big data begins to absorb more and more of the conventional functions of conventional database models. But there are some risks of using big data because you don't use these rigid database schemas, and there's a whole array of, as I mentioned, emerging techniques to minimize the risks associated with using big data analytics. So, I said I like to invent terms. One of the terms I created because there wasn't a good way to describe what's emerging in the big data world, and so I thought of it as a multi-platform ecosystem, and I contracted that into the Pleco System. And there are 4 aspects of the Pleco System that I think become very evident the more you begin to work with big data. So there's service platforms, there's data platforms, there's application programming interface platforms, and there's experience platforms. And I give examples of each of those. There's 4 principles. There's exponential growth across each of those platforms. There's synergy across the different data types and the different platforms. There's liquidity of data, and this comes back to the criticality of being able to federate data from multiple different sources, and hence the argument for more global collaboration. And then there's person centricity and the ability to bring together everything about an individual to be able to do ultimately what we do in medicine and in health is we do the mashup of little data, which is everything we know about you versus everything we know about the human condition, and then we do the algorithmic mashup to see what is it we know about you that matches what we know about the human condition that we can benefit through some intervention. And then the topology of the data and analytics. So one of the emerging themes in the big data world is rather than bring all the data into one location over time, you want to find out what's important, separate the signal from the noise, and do some filtering and bring the analytics to the data so that you don't have to bring every bit of data on everything into a single location. And there was actually a paper just published out of MIT a couple days ago that proved that they have some compression algorithms. So, for example, in DNA, the sequences of DNA across the entire genome are not random. There are patterns that repeat over and over and over again in DNA. And so what they found is that when you try and compute across, so our ability to generate DNA sequences is way faster than Moore's law. I mean, our accumulation of genomic data is just beating the pants off of Moore's law. And so our ability to compute on the data we have is suffering. So what these folks at MIT have done is they've shown some compression algorithms that have basically accelerated the ability to do analytics across massive amounts of genetic data with minimal loss of data integrity. And in driving value from the Pleco System, I mentioned big data versus little data, and there's something I call the innovation index. So that's the density of platforms and platform skills within a social network. So there's the social Pleco System of interdisciplinary skills, and there were several examples of that given today in some of the talks about multidisciplinary approaches to problems, multiplied by the balance of the missionary-mercenary ratio. By that I mean those who are doing research for the pure benefit of improving the health of our communities versus those who are looking for that one thing that's going to make them $1 billion. And there's not a right answer to that, but it's a balance that's important. So one of the things that struck me, my daughter came to me in her high school biology course and said, "Dad, I don't get meiosis. Will you help me understand meiosis?" So like any good father, I went to YouTube and pulled up a video of meiosis, and for the first time I really understood how meiosis really works, because the video was just unbelievably simple. But in the interest of time, let me just say that there's 3 events that happen in meiosis that are utterly mind-blowing. So we all read about it in high school biology and forgot about it. This is one of the most profound inventions of evolution. With the first thing that happens is the sister chromatids align at the kinetochores, and they massively swap. Think of genes as ideas. They massively swap genes between the chromatids. And then moments later, there's random assortment. You get the maternal chromatid on chromosome 1 and 5 and 7 and 9, 10, 11, and you get the paternal in the others. A random assortment. And then minutes, days, or weeks later, fertilization happens, and you get the fusion of the sperm and the egg. So what happens in a matter of minutes, hours, and days is 3 mashups of tens of thousands of ideas into a whole new individual that's never walked the face of the planet before. So there are more viable permutations of the human genome than there are people who have ever walked the face of the planet. Again, another argument for why global collaboration in the world of genome and panorome and exponential and big data becomes critical. But when you think back to meiosis, it's a model for innovation that, again, preceded any of our current human design models. I think one of you had your microbiome sequenced. As I said, I get 4 of them sequenced every 3 months. This is Larry Smarr, one of the pioneers. Now, he did his PhD about the collision of black holes when he was in Stephen Hawking's lab. Then he set up the first supercomputing center in the world. Then he assembled the team that invented virtual reality, and then he got interested in microbiomics. So how does an astrophysicist, supercomputer geek, virtual reality geek end up in the microbiome? It's the same kind of challenge, because it's massive amounts of data, terabytes of data in the microbiome. So these represent the profiles of the microbiome of 6 different people. And each one of these bars is a different phylum of bacteria, and each one of the individual bars is a species. So on average, we have 2,000 to 3,000 bacterial species, many of them identified only genetically, none of them ever seen under a microscope, none of them ever identified in a microscope. And a fascinating side note is that genetic diversity in Africa is greater than any other continent. So is microbiomic diversity greater in Africa than any other continent. But the important thing is that there are 10 times as many bacteria in our gut as there are cells in our body. There's 100 times as many genes in our microbiome as there are genes in the human genome. There are almost 100 diseases already recognized with a very characteristic signature fingerprint of the microbiome and that disease. And we know that there's a very direct interaction between the microbiome, the genome, the immunome, and the neurobiome. And there was a study published recently showing that prolonged antibiotic exposure in children completely rewires their brains. The brain of a kid who was on antibiotics for prolonged periods of time as a child is not the same as a brain of someone who did not experience that exposure. Mike Snyder, chair of genetics at Stanford, has said there's no such thing as a common disease, and that's absolutely true. As I mentioned in one of the Q&As earlier today, Eric Schott at Mount Sinai has shown that there are 5 distinct subtypes of type 2 diabetes at least, and we're going to find out about almost every disease that we think is a single common disease now. Again, the size of cohorts becomes very important. Collaboration becomes very important. So a couple of questions that we're still really clueless about. Why do we have 23 chromosomes? Why not 15? Why not 60? Who knows? Why are some closely related genes functionally co-located while others are on completely different chromosomes? What are all the mechanisms for balancing the need for innovation and diversity, as we see in meiosis and as we see in metabolic fatigue, vis-a-vis the need for conserving valuable genes? So there are several mechanisms that are being discovered about how we protect valuable genes and other mechanisms for how we generate deliberately diversity. And, for example, mitochondrial DNA. So why is it so important that we understand this balance through evolution of how sexual reproduction in the genome has evolved when we now have mechanisms to affect this? So this gene drive mechanism combined with the CRISPR editing tool can allow us to effectively wipe out genes in any species, just wipe them out across the population, a specific allele. We have the ability to do that. We have the ability, as I showed in the video, to select the color of your eyes, coincidentally with selecting out for BRCA. We have the ability to select against a prevalent allele which has some adverse consequences, say sickle cell anemia. Well, we know that the heterozygous state of sickle cell anemia is a genetic deliberate event to protect against malaria. It's the homozygous state that's really, really bad. Well, what if we start knocking out alleles that seem to have some adverse consequence, but they've got a 2% prevalence? Well, why are they there at a 2% prevalence? What benefit do they confer to the population as a whole? How do we know? The question becomes, how confident are we that we really know what we're doing when we knock out an allele with this capability? These capabilities are not sci-fi. They're here today, they're very well established, and they work. And that's why there's already an international agreement about what we will and will not do as a research community with the gene drive technology. So why are aging and the average age of death so tightly controlled in every species and reveal such a wide variation in lifespan? So why do some species in a genus of turtle live for 200 plus or minus 10 years and another live for 20 years plus or minus 5? Clearly there are multiple mechanisms that control longevity, and all of Ray Kurzweil and others' efforts to extend life are trying to essentially override those control mechanisms. What are the implications of that? We really don't know. So what I'm trying to say through all this is there's so much more we don't know that we do know, that in the interest of improving the lot of the human condition, we are taking some unknown risks. So these are just some, I'm going to do a little bit of future casting about the future of sequencing, but just some very quick assumptions. The cost is going to continue to go down. The accessibility of sequencing is going to become miniaturized and distributed. The complexity of interpreting the on average 3 million variations for an individual human from a reference genome is going to get better and better, particularly with some of these compression techniques out of MIT. The precision of our interpretive capacity is going to grow. Another term I invented called genofencing. Geofencing is where, with augmented reality, you know what health food stores and gyms are within say a quarter mile of where you are. Genofencing basically says, here's the bounds, and this is what 23andMe does in their interpretation of your exome. You are at higher or lower risk of this, you're lower risk of this, and higher risk of this. That's genofencing. As we do that, how do we make it actionable and contextual so that we can actually give useful advice? Most people get their exome back from 23andMe and they go, I don't know what I'm going to do about this, but we are going to get better and better at managing that. Ethics, we need to take these issues on very deliberately, and non-discrimination. So there's a big issue here. So 95% of the UK biobank is white, and they are represented with 50% less morbidity than the average in the population. A lot of it socioeconomic, a lot of it cultural. How do we make sure that we're non-discriminatory in how we apply this? I had a conversation with Robert Wood Johnson about this just last week, about how we systematically try and address that issue. So the sci-fi writer famously said the future's already here, but it's unevenly distributed. So there's a lot of things already happening. So the federated data enclaves that I mentioned in credential research, it's already happening. New England Journal of Medicine published a paper recently showing, they looked at all health plans and they showed that social disparities in the outcomes for hypertension and diabetes were much less at my institution, Kaiser-Permanente, than anywhere else. Well, all of our researchers called each other up and said, why didn't you tell me you were doing this? Turns out nobody at Kaiser-Permanente did the research. There's a data enclave that Health and Human Services manages. They include the health plan of the member, and someone outside of Kaiser-Permanente did the research without any of our knowledge and published it in the New England Journal of Medicine. That's what a data enclave and credentialed access allows for. So that future's already here. Sneak peeks, I showed the video about how you can do mashups of potential father and potential mother, and then manage the embryos. Stem cell harvest and CRISPR-Cas9, we can do repair of known problems and then reimplant and eventually, almost for sure, cure diseases like Parkinson's and so forth. Plastic surgery and genetics. So there's already been an attempt in China that's been basically derided everywhere else in the world, where they're trying to do genetic editing in embryos, very, very dangerous and ahead of our knowledge. Novel approaches to complex problems. So autism has really been we know that there's over 600 SNPs associated with autism, SNP variants. It's been very refractory to science. Google by January is going to have 10,000 trios, index case, mother and father, 100% genome sequenced to look at this. But somebody else just published a study that was incredibly creative. They took stem cells from the skin of autistic children of varying severity, and they cultured brain organelles in vitro, because they took neuro stem cells, conditioned them to grow brain organelles, and they found that they were profoundly abnormal. They discovered that none of the principal genes in neurogenesis were abnormal, but the gene regulation was profoundly changed. They looked at the microRNA that was regulating those genes. They changed the microRNA milieu within those organelles growing in vitro, and they generated normal brain organelles from the abnormal stem cells of those autistic children. So they're going to, again, this multidisciplinary look at old problems is going to help disambiguate some of the complexity. We know that a very high percentage of failures in vitro fertilization come from the quality of the egg, the cytoplasmic factors in the egg. Blockchain technology, I mentioned liquid biopsy for early detection. So at the Institute of Cancer Research in London, the Royal Marsden NHS Foundation, they just published a study where they can detect recurrences of breast cancer many, many months ahead of any other evidence by doing liquid biopsy of the blood, looking for genetic markers of breast cancer. Many startups working on this problem. We know that if you look at gene regulation, and if you look at the microRNA and that up and down regulates your DNA, if you look at it once in a day, you've looked at it once in a day. A friend of mine sold his first startup for $200 million. He's doing a new startup looking at RNA signatures, and what he's found is there's massive diurnal variations, predictable in a single individual throughout the course of a day. So there's up and down regulation of the microRNA that up and down regulates our genome. So again, we're barely beginning to understand what this is all about. And the amount of data storage required for that microRNA profiling is huge. And then risk management, we should be able to better target therapies vis-a-vis idiosyncratic reactions. The recent epidemic of narcolepsy associated with the flu vaccine here in the EU is something that we should know how to avoid in the future. I'm going to run through these very quickly and then cut to the end here pretty quick. So what is the future of sequencing across the human life cycle? So pre-fertilization, I mentioned the GenePeaks mashup, which costs about $2,000, and the procedure that they showed you for embryonic selection was about $16,000. 2,000 couples have done that already. Post-fertilization, pre-implementation, are we going to start picking what color eyes? Liquid biopsies to sequence the fetus. There are a number of companies. This whole business model is doing in utero sample of the maternal blood and identifying the full sequence of the fetus. Perinatal sequencing, if it's not already been done while the fetus is in utero, after birth, is becoming more and more acceptable. In childhood, we know that children born from C-section, 5 years later, you look at their microbiome and you can tell whether they had a vaginal delivery or C-section, because the microbiome that starts off their bowel is from the skin of their mother with a C-section rather than through the vaginome. And there's a whole lot of research around that. And then health maintenance and genofencing. How do we target preventive medicine and health based upon the bounds of the genome of an individual? Genome diet matching, a lot of startups are looking at this as opposed to sort of these hocus pocus blood type diets and so forth that have been very faddish. We will know much more about how to match individuals on the whole, I'm blanking on the name of the protein, gluten-free. The whole gluten-free diet fad, there are very few people who actually need to be on a gluten-free diet, but we will know very soon how to match genome with diet. Earlier detection of disease, cancer management with designer drugs, monitoring recurrences, life extension, and throughout all stages of life, the stem cell capabilities. The microbiome, there's so much known about the microbiome. The exponential growth in knowledge about the microbiome in the past couple of years is really mind-blowing. If you haven't looked into the microbiome, I strongly recommend it. It has a profound influence on health. It has a profound influence on obesity. We know that 18 months after a gastric bypass, most women have an abrupt jump in their weight. And the reason is that at about 18 months, no one knows why yet, but there's a radical change in the microbiome of people who have had a gastric bypass, and it's a result of 300 calories of pyruvate and acetate that are generated by the microbiome that's been changed at that point in time. So future questions. How do we address the imminent needs of training the healthcare professionals in genetic counseling? Today we have specialized genetic counselors. We're going to have so much information in 3 to 4 years that every clinician is going to have to become skilled in genetic counseling. How do we deal with the incidentaloma? So we're going to find all kinds of things that might be really significant for your health in some people but not in others. And how do we know if it's an incidental finding or not? We already have that problem with imaging where we find a little nodule somewhere. We don't know if it has any significance or not, but we're going to see that more and more. How do we address the transitional state where we have this exponential growth in knowledge and value of sequencing, but the cost of sequencing is actually stagnated over the past 2 years? It's been on a super Moore's Law decline, and it's been stuck for about 1.5 years, 2 years now. So we're in kind of this never land. And then the whole issue of designer babies. How do we anticipate an imminent awareness of the fact that in Katrina, the Hurricane Katrina, when they did identification through genomics, 20% of the people that they identified genetically were not, their identified father was not their biologic father. In 9/11, it was 15%. In Toronto, across the general population, it's 10%. So one of the consent forms you sign if you do get your genome sequenced is they won't tell you if the father that you thought you had is actually not your biologic father. What are the implications for disclosing our genome and implicating our relatives, because there's a lot you can tell about your family pedigree. How do we foster more international collaboration? And Gil Alterovitz, who I mentioned earlier on the tuberculosis resistance issues, actually did some research looking at co-publication across institutions and found that there was a much higher incidence of collaboration on research and publication if there was cross-fertilization on the governance bodies of those same institutions. And then how do we continuously update everyone about what we learn from big data and how it applies to their little data? So if we tell someone, here's what your genome means today, we're going to know 10 times as much in 2 years and 100 times as much in 4 years as we know today. So how do we maintain? Who does that? How does that work? This is Terra Nova. So what can we do today? Get sequenced, get your relatives sequenced, seek permission to link your pedigrees, because it's a great value to you and everyone, and support an open dialogue around policy and ethics in genomics. So where is this all leading? So my information dense slides are over, so you can relax and take a breath. But there's a couple different alternatives about where this is all headed. So here's what I call homometricus, where we all are wearing these sensors and embedded loops and we're tuned athletes and warriors. That's an idealized version. Here's what more of us resemble as Homo geekus, because we're sitting down too much. And here's what fast food has done for us. I call this Homo fast food or us. And when the flat panel screen was invented, something went terribly wrong. And then there's my view of where we need to go and the evidence. And so one of the talks I like to give is called Using Modern Technology to Restore Ancient Wisdom. The scientific evidence that meditation of all different kinds, so traditional transcendental meditation is not for everybody, but some form of meditation, whether it's prayer or just silent time, it has been proven to be a fantastic benefit. And Jeff Hammerbacher, founder of Cloudera and one of the smartest people on the planet, is working with Eric Schott at Mount Sinai, and they're doing the big data analytics now to get into greater detail of how meditation helps us live longer. But it's clear that a lot of what we need to do, and there was an article in the New England Journal that just basically criticized the precision medicine initiative and all the funding going into precision medicine, when we already know that the disorders of lifestyle, of poor sleep, poor exercise, poor diet, poor maintenance of our healthy, of a healthy social relationships and our expression of love and caring for other people, those are the things that are driving poor health more than precision medicine. So there was a population biologist who basically said, look, why are we spending all this money on this other research? Well, I believe that there's a synergy between the two that we can help, but we do need to do both. And then coming back to Andrew's raising money for the children, the evidence that's accumulated recently for how in utero and early childhood experiences really condition our health span as well as our lifespan for the rest of our lives should cause us all to think about redirecting and reallocating resources from treating people who have 5 comorbidities when we could start preventing all 5 of those comorbidities at an earlier phase. So Dorota Chapko earlier today revealed her PhD thesis that in utero stress drives lifelong disease. Rosie Cornish gave a talk today about how breastfeeding is associated with higher IQ. King's College just published a study a couple weeks ago that shows how early childhood stress causes remodeling of gray matter in the brain. So they did MRI scans in teenagers who had been followed since early childhood with stress and showed that areas associated with depression and anxiety had maldistribution of gray matter that was characteristic of those 2 disorders. That was a result of that early childhood stress, modifying the microbiome through antibiotics, the C-section results and so forth. So the question is really, how do we reallocate resources where we have a greater impact and impact health span and lifespan in early life through better coaching for parenting, better prenatal health, better perinatal health, better support for young parents who are struggling to make a living at the same time they're trying to raise their children. So finally, Hubert Humphrey, one of our former vice presidents across the pond, has a saying that's up in the Department of Health and Human Services. It says, it's been said the moral test of government is how that government treats those who are in the dawn of life, the children, those who are in the twilight of life, the elderly, and those who are in the shadows of life, the sick, the needy, and the handicapped. And so I think all of us in how we use technology are really obligated to not just think about how we solve this micro problem in front of us, but how we apply that to a population as a whole and how we focus more of our resources on parenting and our children to really improve the health. And lastly, Maya Angelou, I think, said it best. I've learned that people will forget what you said, people will forget what you did, but people will never forget how you made them feel. So as we do our work and as we collaborate with others, remembering the golden rule and remembering that our goal, after all, is to help the health of our communities, and that includes the people we work with every day, I think that we can be consistent in our goals, our motives, and our values throughout our personal life and our professional life. And that's it. There's just some references, and thank you very much.